welcome Owen Brody here, a senior scientist at LBL, Deputy Director of the Climate and Ecosystem Science Division, and also leading a number of very large, really large-scale projects, including the Earth Microbial Engines Grand Challenge, the Joint Berkeley Initiative in Microbiome Science, so on and so forth. And his work is really centered around microbial ecology and biogeochemistry, but spanning from molecular mechanism to entire ecosystems, and again, going across field-based to lab-based to computational models. So to me, it was really the perfect fit to start this session where we are trying to link sequence to function in the environment. And with that, I will just remind you briefly to please use the Q&A function, which is on the right side of your screen, if you have any question during the talk, and I will now hand it over to Owen.
Great, thank you, Simon and Tanya. First of all, can you hear me and can you see my slides okay?
Yes.
Super, thanks. Well, thank you very much for the invitation. First of all, I want to say thank you to JGI. I really appreciate the support from JGI over the years, not just in the production of data, but really deep collaboration in the processing, interpretation, and the science underlying that data as well. And it's a collaboration, I think, that's gone from strength to strength over the years. So many thanks for that. Today, I'd like to talk about approaches to using trait-based approaches to understand the ecology of soil microbes. And I'll give specific examples and a case study of how we're using trait-based ecology. I want to talk about how we tackle the complexity, particularly of soil microbiomes through this approach, how we infer microbial traits using first principles approaches from bioinformatics, how microbial fitness traits are important for soil organic matter formation, how we then use models to integrate traits, their trade-offs, and environmental filters, and then some approaches to begin scaling trait distributions. And as I start, I want to first mention that this work is performed by the folks you see here. I'll introduce them as we go through. And really, I'm a spokesperson for a team or a number of teams of very talented people. So I want to start off with some context, and that's, as we know, the terrestrial carbon pool is not in equilibrium with the atmosphere. In other words, there's more carbon in soils and in plants than there is in the atmosphere. In fact, 3 times more carbon in soil than the atmosphere, and 4 times more carbon in soil than in vegetation. And the reason for this disequilibrium are these what you call dissipative structures over here. So both plants and microbes work together to take carbon dioxide from the atmosphere, build structures that combined, the interaction between these structures enhances the dissipation of energy through things like evapotranspiration and respiration and things like that. These interactions are central not just to the carbon cycle, but to the energy budget of the Earth system. So understanding plant and microbial interactions is key for a number of reasons. Now, soil organic matter formation is key here. We now know that microbes are key to the formation and stabilization of soil organic matter. My colleague who is now at Livermore, Noah Sokol, his work with Mark Bradford, introduced a really important, and that's the location in soil where organic matter enters is important for its stabilization. And Noah, in his work, identified particularly a contrast between rhizosphere and what you might call bulk soil. So areas adjacent to plant roots with high microbial density are extremely important for the formation of stable mineral-associated organic matter. In other words, carbon coming from a plant going into regions with high microbial density, microbes producing other compounds, producing biomass, and then those materials being stabilized on soil minerals, as opposed to a leachate, for example, coming into the soil and directly attaching to soil minerals. This appears to be less stable than the microbial route. And we're going to talk a bit more about that. So the rhizosphere versus bulk soil is important. We know that microbial carbon use efficiency, otherwise known as growth yield, for example, is key to soil organic matter formation. But it's an emergent property. It's not predictable from individual variables. It emerges from a combination of multiple factors. Soil organic matter formation is correlated with carbon use efficiency. This is from Cynthia Collenbach's work, empirical evidence showing COE and soil organic carbon deposition on minerals. We also know that the substrates matter. So the chemistry that microbes are using, the carbon chemistry, particularly the oxidation state of carbon, that strongly impacts growth yield, and therefore it affects carbon use efficiency. And we know that the properties of the microbes themselves are extremely important. And you heard from Daniel Segre this morning in his work with Mustafa Safieddine showed that the potential carbon use efficiency across a range of genomes is really related to the types of organisms. So it has a phylogenetic signal, and that phylogenetic signal is underlain by differences in metabolic network configurations in different organisms, and things like genome size and GC content are also related to potential carbon use efficiency. So microbes and substrates are important, and I'm not going to talk about minerals today, which is also an important process here. Now, to make sense of this, we have a big challenge, though, and that's because soil is the most diverse, maybe arguably, microbial ecosystem in terms of number of species per number of cells in the system. And that makes our challenge difficult to understand, and couple that with the fact that we have not cultivated most organisms from soils. So to deal with the complexity, trait-based ecology is really an attractive approach. When we talk about traits, we're really talking about things that features of organisms that explain differences in fitness among individuals. They can be simple, like the presence of a gene, or complex, the entire architecture of a metabolome, or things like the optimum growth rate of an organism, for example. So traits across different scales. By assigning traits or identifying traits of organisms, we can then start to reduce dimensionality. In other words, collapsing many genes into pathways, and pathways is into classes of metabolism. We can start to reduce the diversity of traits into a representation that's either that's adequate to be able to explain patterns that we observe. And that brings me to the next point here. Is really a goal of trait-based ecology is to provide mechanistic explanation of the patterns that we observe, whether those are ecological or biogeochemical, but also to identify generalizable combinations of traits. So how do these traits come together? How do their trade-offs impact the fitness of organisms within their system, and how do they change across scales? This is then the basis for building maybe predictive capabilities through models of how these traits interact within populations, communities, and how the local context, the environment, or the constraints on the activity of organisms, how that impacts the patterns that we are aiming to explain. So soil microbes or microbiomes are complicated systems. They're not infinitely complex, but there are key traits that are necessary for microbes to be able to survive, persist, and prosper in the soil environment. Traits related to accumulating resources, how they use those resources, how microbes deal with stress, for example. These are all important. And of course, there's a zero-sum game, so there's trade-offs. Investment in one type of trait means that there's less investment in another type of trait. So these are manifested as trade-offs due to laws of conservation of mass. Microbial traits, though, we also know that there are phylogenetic signals in both the trait linkage, and therefore those phylogenetic signals also lead to phylogenetic signal in the response of things like stress. So certain types of microbes responding positively or negatively to a certain stress or a certain stimulant, and that's because the distributions of these traits is not infinitely complex. There are evolutionary constraints, so that works in our favor. And we also have environmental filtering. So environmental filters constrain the realized niche. So microbes might have a fundamental niche that they could inhabit, but the environment doesn't allow that. So there's a constraint also on the realized diversity through the constraint on how niches come together. Okay, so how do we do this? Bear in mind, we can't grow most of the organisms that we may care about in soil. So our lens on the microbial ecosystem now is increasingly through things like high throughput sequencing. That gives us our blueprint of microbial metabolism. That, of course, is a genome. It's a hypothesis at that point. We need to ground truth that. So there's still a really important role for isolation and benchmarking and verification of the inferred traits that we get from microbial genomes. But then in order to scale that information, we also have to consider other things like mineral interactions, microbe interactions, mineral interactions, how microbes function in porous media, how diffusion affects the availability of substrates and interactions, how plant metabolism in the context of soil can create distinct compartments and niches with very different chemical and physical properties. and then how plants differ in their traits that impact microbial assembly in the rhizosphere, how landscape constraints impact the availability of water, energy, and plant species distributions, and then, of course, how global forcings, large-scale climatic variables influence functioning at the landscape scale. So it's a big challenge, but we have to start somewhere. So we've been working, this is work from Ulas Karawas, who's been developing an approach based on hidden Markov models to take genomes and distill genomes into traits. Now, if you want to read more about this, I don't have time to talk about the details today. This is available as an R package on GitHub at the link here, and we'd be happy to talk you through the use of that. Distilling genomes into biophysical, life history, resource acquisition, stress response, and energy generation traits, amongst other things. This allows us to build a picture of the lifestyle of a cell. So things like estimating cell size, surface area to volume, which allows us to get at things like transporter density, mu max, the minimum generation time of an organism, how fast does it grow, what resources does a microbe use, and how does it invest its genomic and proteomic resource in acquiring those substrates, how microbes allocate energy across their processes and stress response and things like that. So Ulas has been analyzing all bacterial genomes from soil, and here's an example of something that emerges from that. Look at the relative genomic investment organisms that you find in the rhizoplane and the rhizosphere. They are particularly enriched in carbohydrate transport. Maybe that makes sense because of the abundance of fixed carbon in the root zone. I think that probably does. What you can also do then is reduce the dimensionality. So you can take all the genomes, cluster them based on their traits and the trait linkage, and then collapse the genomes into guilds. So these will be organisms that perform a specific function or inhabit or have greater fitness in a specific niche. Just examples here, these organisms here, because they've got a higher genome investment, they're enriched in investment in transporters versus these ones that are enriched in investment for polymer degrading enzymes. You might be able to classify these as polymer users, exudate users. It may be more complicated than that, but that's as an example. And then what you can do is you can extract trait distribution. So we can measure things like the minimum generation time, which is basically mu max for your Monod or Michaelis-Menten models, and contrast exudate and polymer users. We can look at genome size differences, for example, and estimate how genome size might scale with cell volume, for example, and then predict things like optimum growth temperature. These are some examples of things that we can predict. So with that, I want to talk you through an example of how we try and take observations and put all those observations together, develop a model that allows us to calculate how traits interact, how trade-offs are manifested under different conditions, and how they contribute to emergent carbon use efficiency observations. And I want to start off with some work that was carried out by Kate Zalnina. This was in collaboration with Trent Northern, and you've heard about Trent's amazing metabolomics capabilities. And so we collaborate together, and working with Mary Firestone and other folks, we're able to show in the paper published a few years ago that root exudation chemistry and microbial substrate preferences were very strongly related when we looked at patterns of rhizosphere microbial assembly. And what I mean by that is that when we look at the genomes of microbes that respond either positively or negatively to a growing root in soil, that we could see differences in microbial substrate preferences predictable from their genome. When Kate also looked at the exudation pattern of plants over time during development, we could see corresponding differences in the types of compounds that plants were secreting as root exudates as they grew. And the combination of microbial substrate preference and the chemistry that the plant was releasing as exudates over time did a pretty good job of explaining the patterns of microbial community succession. Now, there's more to it than that, and I want to talk through a little bit more of what we found since then. Just to summarize Kate's work, the genomic traits associated with soil niche, and focusing here on the organisms that appear to have a greater fitness in the rhizosphere. Rhizosphere adapted organisms, in this case, to our surprise, had slower maximum growth rates. So their minimum generation times were longer, meaning generally slower growing. They had fewer ribosomal RNA operons and less codon usage bias. So we have two genomic proxies to be able to confirm this observation. They also had a higher number of monomer transporters for exudate compounds, particularly organic acids, and then a lower genomic investment in extracellular enzymes, particularly glycoside hydrolases. So suggests that they really occupy a different resource niche. Now, Kate, on the other side of this, the bulk soil bacteria, these are the ones that didn't respond as well to growing roots. We could see that those bacteria invested more in carbohydrate active enzymes and actually secrete about 10 to 20 times more when you test that in the lab. So they have potentially a greater metabolic cost in producing and secreting these proteins that might impact their competitiveness and also their carbon use efficiency. So in order to represent this knowledge, these traits, our assumptions of their trade-offs or estimate their trade-offs and how environmental filters interact in this sort of schema here and how that impacts carbon use efficiency, Gianna Marshman in our group, building on work from Jinyun Tang, has been developing a dynamic energy budget model, and we're using genomic information here to parameterize this model. The model represents the allocation of resources in a microbe through reserve into growth and maintenance, etc. So things of that cost to microbe, such as maintaining itself, producing extracellular enzymes, etc., will all trade off against growth. And then the uptake of substrates, there's costs associated with that in terms of allocation to transporters, etc. So we're going to explore how this comes together. So what this looks like in terms of an energy budget allocation is where we have carbon use efficiency will depend on properties of the substrate. So the free energy available to the substrate, how the delta G of that substrate contributing to ATP production, and also the stoichiometry of the substrate. That influences the efficiency of how these substrates are assimilated into reserves or assimilation efficiency. Then we look at the protein yield in generating structural biomass. The protein translation rate and protein efficiency has an allometric scaling, in other words, depending on the size of the cell and the allocation of proteins. And I'll show that in a second, but also it's impacted by ribosomal RNA copy numbers, which has an impact on efficiency. And then calculating the fraction of the energy budget that's allocated to maintenance, as I said, and things like extracellular enzyme production, as opposed to structural biomass. That would reduce growth efficiency and carbon use efficiency. So this builds on work from Jinyun Tang, and Jinyun developed this approach called ECA kinetics, equilibrium chemistry approximation kinetics, and that allows us to be able to calculate the effective affinity of transporters. As an example, when multiple substrates are competing for those transporters, it also allows us to reflect the affinity of a specific microbe when multiple microbes are competing for the same transporters. In this case here, the uptake of substrates by a microbe is diffusion limited. We know that. So the surface area of the cell is important to increase the area for diffusion. But transporters, so the actual area of the surface that is involved in uptake is really important. So transporter density, and then of course the substrate properties through their energetics, and assimilation yield is also important in determining how much a substrate an organism needs to take up to build biomass. So I think I've said most of this already, but cells and substrate size and diffusion, they define microbial uptake. Monomer uptake is limited by accessible microbial transporters. And what we're doing here is we're allocating transporters based on cellular demand. So depending on the optimum growth rate of an organism, which we can infer from a genome, and then the yield coefficient for specific substrates, we can then define what the transporter density would need to be on the cell surface to be able to reach the optimum growth rate. So because now having established what the optimum transporter density is for a type of organism, what we do is we allocate those transporters to different substrates according to their genomic allocation in the organism involved. And that looks something like this here, where we can predict according to cell volume, the relative biochemical composition of a microbe, how much DNA it has, how many ribosomes it has. That gives you your protein synthesis rate. What are its investments in membrane, et cetera, as well, and other structural components. So we can get the cell membrane fraction, protein synthesis rate, et cetera, as well. And all of this is towards an objective function of reaching the mu max, or the minimum generation time. Now, there's a trade-off there in protein synthesis efficiency. This has been observed empirically, and we've built this into the model here as well, where ribosomal RNA operon copy number, that there's the more ribosomal RNA operon copy numbers you have, the lower your protein efficiency. So although there's a faster growth rate associated with this, there's a trade-off with protein synthesis efficiency. And then to calculate the yield based according to different substrates, we're using the KBase app from Hyunsub Song, the thermostoic wizard here. Okay, and then our transporters are predicted and estimated using TransportDB, and we allocate those according to the genomic investment in those transporters. Okay. So Gianna then took 83 root metabolites that we had measured from Kate's work and 39 isolates that we'd sequenced, then we'd estimated all these traits, and then did simulations of where and when traits are important and what trade-offs we might expect. One of the first things that emerged here is that transporter density is really independent of cell size or surface area to volume relationship. It could be surprising because diffusion should be a linear function of cell surface area. But what happens here is that smaller cells, although they have larger surface area to volume ratios, they have a higher relative investment cost in transporters. And so that limits transporter density. And so that expected relationship sort of breaks down because of a trade-off. We can also see that the uptake kinetic parameters have really strong correlations with transporter density. So I mean, transporter density is going to be a driver in investment in transporters, a driver in uptake kinetics, but surface area is important for transporter density. And we can see that trade-offs as a result of this between maximum specific uptake rate and half saturation constant. So the concentration of a substrate at which microbes can achieve half of its maximum uptake rate, that they trade off. In other words, as the uptake rate gets higher to support a faster growth rate, the organism needs to persist in an environment with higher substrate concentrations. And we can see the affinity, so that means the available area for substrate uptake is influenced by surface area to volume ratios. But when we get to above a certain affinity, there's really little gain in transporter density. So you see the transporter numbers dropping off here at that stage. Okay. So looking at what this means for things like substrate uptake, Vmax, and half saturation constant and affinity, we could see that surprisingly, the negative responders in the rhizosphere have higher maximum specific uptake rates, or predicted higher specific maximum uptakes, and higher affinities, but they also have higher half saturation constants, except for organic acids. And what that means is that the organisms that are responding positively to root exudates, they have lower half saturation constants. In other words, they are more competitive for the substrates in the rhizosphere, specifically for these substrates in the rhizosphere, at lower concentrations. And these concentrations are actually consistent with measurements of these substrates in the rhizosphere, meaning positive responders are tuned to the substrates concentrations in the rhizosphere. This allows us then to make predictions about how different substrates will impact the carbon use efficiency of each of these groups of organisms in the rhizosphere. So when we look at those predictions of carbon use efficiency from batch simulation, so feeding groups of microbes, feeding all these microbes different types of substrates individually, one of the first things we can see here is that carbon use efficiency for the organisms that respond positively to root growth is higher for amino acids, organic acids, and sugars, meaning they're going to create more biomass per unit carbon substrate that they're taking up for these substrates. And so we see that as we hypothesize, there's a significant association between organism type, how they respond to the rhizosphere, and carbon use efficiency for these substrates. That has implications for the formation of organic matter, for mineral-associated organic matter, as I'll show. One of the key variables when we do random forest analysis of what variables underlie these observations is that the assimilation yield, and that's going back to the thermodynamics of the substrate that the microbe is taking up, that's really important in defining carbon use efficiency in general, but also ribosomal RNA copy number and the kinetic parameters that we just predicted. So the half saturation constant, the maximum specific uptake rate, as related to transporter density. Now, the reason why these microbes have higher carbon use efficiency is both the substrates that they're using, but also differences in protein synthesis efficiencies, because the positive responders, they have slower growth rates, they have lower ribosomal RNA operon copy numbers, and therefore their protein synthesis efficiency is actually higher. So they have less of a trade-off, and in some cases, not all cases, they're consuming substrates that have a higher thermodynamic yield. So these factors come together and influence the carbon use efficiency of these organisms within the rhizosphere. That's a really important finding. Okay. On the other side, what we saw was that the production of constitutive production of enzymes by these negative responders who don't really like the rhizosphere as much, that was less important in determining carbon use efficiency than substrate availability and protein synthesis efficiency. So we can compare and contrast. Okay. So I want to summarize this section, and then I give a sort of a brief overview of what we're trying to do next. Carbon use efficiency is important as a control in mineral or socioorganic matter formation. And the CUE in the rhizosphere depends on an interaction between substrate-specific and microbial population-specific factors. And now if we come back to something that Noah had proposed, is that you've got two pathways of organic matter formation, mineral socioorganic matter formation in soil. You've got the microbial pathway where the substrates go into microbes before they get onto minerals. Microbes process them and result in more stable organic matter formation versus the direct sorption pathway where substrates go directly onto soil minerals. We can now start to make some predictions, which we can test in experiments. And that's the substrates with low free energy, so low yield, are biosynthesized with low efficiency. And that might reduce microbial assimilation, leading to more of the direct sorption pathway. Another hypothesis might be that organisms and the use of substrates with higher free energy that result in higher carbon use efficiency, they're under selection in distinct soil compartments because they improve the efficiency of microbial biomass production. And we might expect to see those substrates enter into soil organic matter formation, soil organic matter through the microbial pathway. And I already said this: constitutive enzyme allocation in bulk soil, adaptive bacteria is not a major influence on CUE in batch simulations. And Noah has designed and run an experiment to test this, the DripSip experiment, looking at interactions with drought. And we're excited to compare these hypotheses with observations fairly soon. So going forward, I just want to say that we're very excited to work with KBase now through the Microbes Persist Science Focus Area that Jennifer Pettridge leads at Livermore, to integrate multi-omic phenotyping, including quantitative SIP, which really is important to give us in situ observations of microbial growth and death under different conditions with different substrates. With the microtrait trait inference pipeline and the DEB microtrait model, and Ulash and Gianna will be building components of this over the next year to 18 months and collaborating with Bruce Hungate and the folks at Livermore to build in the multi-omic phenotyping connections into microtrait. We're excited about that. I also want to say that the last slides that I showed really were representing batch simulation. So feed a microbe, a substrate, see what happens. The reality is that the substrate concentrations and types change over time. So Gianna has started tackling this by simulating dynamic root exudation. She's developed, or she's modified, an existing dynamic energy budget model of the plant that does the same thing. It represents resource allocation above and below ground, driven by microclimate data and plant photosynthesis model. And then we can constrain that by our knowledge of what exudates are released when. So this drives our exudation rates, and then our knowledge of the chemistry of exudation allows us to have dynamic chemical exudation from roots over time and use that to then drive our microbial model. So Gianna, just about finished. Yeah.
Okay, perfect.
Okay.
Thank you.
So Gianna has been driving the DEB model with a root exudation. This is an example of root exudates in different chemistry, and is able to show how structural biomass of bacteria changes with dynamic exudation. And some initial observations for this, we can see still the higher assimilation rates for the positive responders, and carbon use efficiency has some very interesting temporal dynamics, as well as dynamics between different types of bacteria related to patterns in root exudation. And I'll be happy to talk about that later. If I have time, Simon, I have 2 slides on scaling, but I could leave them out and bring those up in discussion. What do you think?
Yeah, I think it's probably better because we do actually have some question that, I would love to take at least some of this live, if that's all right.
Okay, great. Yeah, I'll skip to the
you.
last slide and I'll go to my acknowledgements on this.
Perfect. Thank you. So
And
yeah,
thank you. Okay.
Thanks a lot for like such a great talk and such an impressive body of work. Going straight into the question to try to get some of this live. The first question was about the type of soil, so mineral, organic, mixed, disturbed in natural and disturbed systems, and different soil layers, and how this impact microbes with respect to carbon use and functional traits. Can you talk a little bit about that?
Yeah, that's a great question, and sort of I vaguely alluded to that in the beginning when I said minerals and porous media and all that is kind of important. The soil type the one we're working on here is a sandy loam, but clearly the soil texture, the sand, silt, and clay content, that affects that's underlain by differences in chemistry of minerals, which affects the absorption of substrates onto minerals. That affects the structure of the soil, the porosity of the soil, the diffusion of substrates to microbes, the ability to hold water. All of that is super important. And then even within a soil type, as you go through soil horizons with depth, all of those things change. So to be able to build a model that represents a soil profile, for example, you have to incorporate all of those variables as well. So physical, chemical, biological processes are extremely important to that. That's really important and great question.
Sounds good. Second question, I should have said like the first question was from Laura. Second question from Adam was, how do microbes with good CUE impact nitrogen using the system? So basically it's good CUE and nitrogen.
Yeah, good. That's a good question. And actually you could almost flip that question as well. How does nitrogen impact CUE in the system? So the stoichiometry of the substrates that the microbes take up relative to their own biomass stoichiometry is really important to carbon use efficiency. So if microbes have insufficient nitrogen to build their biomass but plenty of carbon, then you have overflow or waste metabolism, and then you end up with low carbon use efficiency. You add in additional nitrogen or compounds with a lower carbon nitrogen ratio that's more similar to the microbial biomass itself, that will impact carbon use efficiency. The carbon use efficiency, let's say, if you compare between, as an example, maybe fungi and bacteria, where fungi have a higher C to N ratio of their biomass relative to bacteria, fungi will have a lower per unit biomass demand for nitrogen. Therefore, the nitrogen uptake rates may be lower, for example, than in a system that might be dominated by bacteria, for example. Now, they're very simple generalizations, but hopefully that gets at the essence of your question.
Yeah, and I'm sorry